So hello everyone and welcome to our afternoon Q&A session. My name's Jessica and for today's Q&A I am joined by Stephen Blakemore, who's Vice President Translational Oncology and Diagnostics at Bicycle Therapeutics, Mike Fisher, who's Vice President and Head of Business Development at Oncommune, and Thomas Condamine, who's Senior Director Translational Scientists at Charisma Therapeutics. Please do leave any questions you have for our speakers in the discussion wall on Swapcard, and all our speakers are live and we'll be happy to integrate these into the discussion. So to get started, Stephen, we'll start with you. So what is the ratio of Nectin-4 to C137 binding bicycles in BT7480, and why did you choose that ratio for the clinical candidate?
Sure, I'd be happy to talk about that. So in the clinical candidate for 7480, we have one Nectin-4 binding bicycle and two CD137 binding bicycles. So it's a 1:2 ratio of the those binding bicycles, or 3 bicycles in total, as I said, 2 CD137 and 1 Nectin-4. And we did a lot of SAR around each of the modalities, going up through 2-to-1s either way, 1-to-3s, and we've essentially found that there was significant benefit in terms of receptor agonism for CD137 by having 2 or 3 actually CD137 binding bicycles, but the benefit between 2 and 3 wasn't really sufficient for us to bear the increased complexity of the chemistry there. So we thought that there was a very significant benefit over 1-to-1, so that's why we netted out on a 1-to-2 ratio.
Great, thank you. And next question for Mike, please. So what do you consider the main benefits of utilising autoantibodies as your approach in diagnosis / in drug discovery as well?
I think there's a few benefits for them. One of the major ones, and that makes people smile when we're talking about doing projects, is the volume of serum or plasma that we use. For an immuno-oncology study, we usually take about 1 microliter. We ask for about 50, but we only actually take 1 out of that and then dilute it by 100 to then run, we'll measure 1500, binding to 1500 antigens from a 1 microliter sample. And then the other one is the stability of these antibodies, because we don't need any special phlebotomy methods that we don't have. We can take a blood sample, leave it on the bench for the day, spin it down, and it's fine. And we've even measured samples that are over 10 years old that have been obviously frozen, but they're over 10 years old, and we're still measuring them. So they're actually very easy biomarkers to measure, and you can measure them by ELISA, you can measure them by Luminex, or even lateral flow devices if you're only measuring a couple of them. So.
Perfect, thanks. And Thomas, just had a question through for you. So are specific monocyte subsets, e.g. classical, non-classical, etc., selected for CAR transformation?
A very interesting question. So we actually don't select specifically monocytes. We pull down all monocytes using a CD14 process, which most likely going to pull down efficiently classical and intermediate monocytes. The likely subset that would be excluded from the manufacturing process is the non-classical, which are CD14 low and most likely not going to remain after pull down purification.
Great, thank you. So Stephen, bringing it back to you, what are the next steps for the research you presented and for that sort of bicycle pipeline?
In terms of our next steps, we'll clearly be reading out more of the biomarker data from the clinical trial, which we'll be doing in the future at academic medical conferences in the future, and adapting our selecting the recommended phase 2 doses and then expanding that in the expansion cohorts.
Great, thank you. So Mike, bringing it back to you. So you obviously talked about a few therapeutic areas in your talks, so oncology, autoimmune and infectious diseases. Do you have plans to expand into any other therapeutic areas, or do you think these are the areas that really have the most benefit in terms of using your technology for them?
Yeah, I think where we are is probably in the right 3 spaces. Immuno-oncology, you're stimulating the immune system to fight the cancer, and therefore measuring that immune system is the sensible thing to do. Autoimmune disease, the autoantibodies are the cause of the disease, and therefore characterizing them is the right thing to do. Again, in infectious disease, all these COVID and other viruses do stimulate the immune system. So that's where we're focused on diseases where it's immune stimulation that causes the disease. So we're probably not going to move into some other areas that where it's probably not sensible to do. But so I think they're the three right ones for us to be working in.
Great. And so, Thomas, bringing it back to you. So cell therapies generally use lentiviral vectors for engineering of cells. Could you comment on the integration and persistence of the transgene using the AD5F35 vector?
Sure. So lentiviral are very well known, as you mentioned, for T cell. However, it's very challenging to transduce macrophage with these vectors. So we had to resort to using an adenoviral vector. They don't directly integrate in the genome, so we don't have problem of integration. They're localized in episome, and the expression is actually quite stable compared to other non-viral technique to transduce cells, with an expression that can last up to several months.
Perfect, thank you. Mike, bringing it back to you, can you give us some more information about the infectious disease panel and the potential applications of this?
Yeah, so we're at the moment we're studying COVID-19. It's about 750 antigens in the panel. So we're going to be looking at about 3,000 patients to look at the differences in response, because as you know, some patients can get the infection and basically be a carrier. They don't have any symptoms, whilst others end up in ICU on oxygen and ventilation. And we don't really know why there is this big chasm between the way that people respond. So we're doing that study as we speak, but we're also working in looking at vaccination, monitoring the change in the immune response to the vaccination. We've also had some discussions with some groups around other viral infections. So, for example, Ebola has some sort of long-term autoimmune diseases that it stimulates. So we're having a few discussions about whether we could look at that and figure out what it is that's causing those, I think it's uveitis is one of the things that Ebola can cause. So we're interested in looking at multiple different sort of viral infections and studying those immune responses to them. So, yeah.
Great. And Thomas, I'm going to go back to you again. So do you expect any on-target off-tumor toxicity as HER2 can be expressed at low levels on normal tissue?
Sure, I can take this one. So we don't necessarily expect any on-target off-tumor toxicity. We're using an scFvDI from trastuzumab that has been in the clinic for a very long time now. The toxicity profile of the antibody is very well known and very well tolerated in patients with multiple indications, especially gastric and breast, where it's approved in the US. And in addition, we don't have a good in vivo model to test this hypothesis with our product. However, we did a number of studies using normal tissue expressing HER2 in vitro. And so far, we've not seen any level of activity of our macrophage specifically targeting normal tissue expressing HER2. So most likely no toxicity. However, the first in human is still ongoing, and we'll have to confirm that during that study.
Perfect. And Mike, it's back to you again. So what are the top 3 takeaways you would want the audience to have from your presentation?
I think the fact that it's a relatively new biomarker class, and they're complementary to a lot of the other biomarkers that are already being measured. So we can correlate, our autoantibodies with other, proteomic approaches, etc. They're easy to measure from a small volume of serum or plasma, and there's a lot of utility there. I think, in immuno-oncology, we've shown that you can predict adverse events, you can predict response. In autoimmune disease, it's useful for characterizing the disease, and again, in infectious diseases, understanding the biology behind what the virus is actually doing with the immune system, I think, are all excellent, ways of utilizing this new biomarker class. So, and we're here to collaborate. If anybody's interested in working with us, we're definitely open to that. So.
Perfect. And we've just had another question through for you, Stephen. So can you talk a little more about the benefits of renal clearance versus liver for the bicycle assets?
The advantage for the bicycle assets through renal clearance, is that we can get rapid removal of the bicycles through the kidneys very rapidly, much quicker than we can through the liver. So it reduces the risk of drug-drug interactions as well.
Perfect. So I think that's all the questions that we do have through at the moment. And if anyone watching does think of another question for any of our speakers, please feel free to reach out to them on the Swapcard platform. And if you have another question for Mike specifically, OncoImmune do have a specific page in our partner section of the platform where you could book a meeting with them to discuss in a little bit more detail. Thank you all so much for joining for the Q&A and for your talks earlier. And I hope you enjoy the rest of the event.
You're welcome. Bye-bye.
Thanks very much.
Thank you very much.
Thank you. Bye-bye.